What is genetic engineering? Genetic engineering is a modern science technique in which scientists change the genes of living things to give them new abilities. Scientists choose and isolate a useful gene from one organism, called the donor organism, and place it into another organism, most often a bacterium. A gene is a small part of DNA that works like an instruction to make a particular protein or trait in an organism, which play important roles in both the structure and functioning of the body. In genetic engineering, a useful gene from one organism called the donor is taken out and put into another organism, bacterium. This new organism, which now has a foreign gene, is called a transgenic organism. The foreign gene works inside it and makes the desired product, such as a medicine, enzyme, or vitamin. For example, scientists can put the human insulin gene into bacteria, so the bacteria can produce insulin for diabetes patients. Genetic engineering is used in medicine, agriculture, and industry to make useful products more quickly and in large amounts. Role of bacteria in genetic engineering. Bacteria play an important role because they are simple microscopic organisms, easy to grow and reproduce very quickly. They do not have a true nucleus, so their genetic material is free inside the cytoplasm, which makes it easy for scientists to reach and work with their DNA. The main genetic material in a bacterium is a single circular chromosome. Along with their main chromosome, bacteria have extra small circular pieces of DNA called plasmids. These plasmids are separate from the main chromosome and can carry extra genes. These plasmids can carry foreign genes and act as carriers, also called vectors, to transfer useful genes into bacteria. Scientists can remove these plasmids from bacteria without harming them, attach useful foreign genes to them, and then put them back into bacterial cells. In this way, plasmids serve as carriers or vehicles vectors for transporting foreign genes into bacteria. Another advantage of using bacteria is their very fast reproduction rate. Some bacteria can divide into 2 cells in as little as 20 minutes, allowing the desired gene and its protein product to be produced quickly in large amounts. Introduction of gene into bacterium. The process of introducing a gene into a bacterium begins with identifying and isolating the gene that codes for the desired protein from the donor organism. Special enzymes, known as restriction enzymes, are used to cut out the gene from the donor's DNA. In the same step, scientists take a plasmid from a bacterium and use the same enzyme to cut it open at a very specific point in its DNA loop. Because the same enzyme is used for both cuts, the ends of the donor gene and the plasmid DNA match perfectly, making it possible to join them together. The donor gene is then attached to the plasmid, forming a new combination of DNA called recombinant DNA. This recombinant plasmid is then inserted back into the same type of bacterium from which it was originally taken. The bacterium that receives and keeps this recombinant DNA is called a genetically modified bacterium, or GM bacterium, and is also referred to as a transgenic bacterium. Once the GM bacterium is created, it begins to grow and divide. As it reproduces, it forms a colony of many identical bacterial cells. Every bacterium in this colony contains a copy of the foreign gene. Under the instructions of this inserted gene, each bacterium starts producing the protein coded by the gene. Because bacteria reproduce so quickly, the protein can be produced in large amounts in a short time. In real-world applications, scientists use this method to produce important proteins such as insulin for diabetes treatment, enzymes used in food production, and proteins used in making vaccines and other medicines. In this way, genetic engineering with bacteria has become a powerful tool for improving health, industry, and science.